Hello and welcome to this afternoon's Biotechny webinar. We're very pleased to have Dr. Kapil Bharti with us today. Dr. Bharti is a Statman investigator in the Unit of Ocular and Stem Cell Translational Research at the National Eye Institute, and he'll be discussing his research into the eye's retinal pigment epithelium and the damage caused by its degradation. Specifically today, he's going to be discussing the development of regulatory paths that have channeled candidate autologous iPS cell therapies into phase 1 clinical trials. We'll be starting the webinar in just a moment, but just before we do, to say that there will be an opportunity to put your questions to Dr. Bharti at the end of the presentation. So if you do have a question during the webinar, to be able to ask your question to Dr. Bharti at the end, please just type the question into the question section of your GoToWebinar window, and we'll select you to ask your question directly to Dr. Bharti after he's finished his presentation. So without much further ado, I'm very happy to introduce you to Dr. Kapil Bharti.
Thank you, Sarah. I want to start by thanking Biotechny. In particular, I want to thank Sarah, Scott, Joy, and Mark for putting together this wonderful opportunity for us to discuss some of the ongoing work that's happening in the lab here. Especially, I will be talking today about developing an autologous cell therapy for using induced pluripotent stem cells. And as part of that process, I'll be kind of focusing on the regulatory path that we're trying to take towards a phase 1 clinical trial in terms of developing this cell therapy. The disease that we, oops, not advancing. Okay. The disease that we focus on is called age-related macular degeneration, short form AMD. As you can see in this panel, people with AMD have a dramatic loss of their central vision. So you can see on the left side how normal vision looks like, and as compared to that, in AMD vision, the person with AMD do not see anything in the center part of their vision. So anywhere they would look inside a room or anywhere else, they won't see anything in the center part of vision. The reason is they're completely blind in that part. The disease has a huge occurrence. There are about 30 million people worldwide that suffer from this disease. It mostly affects elderly people. It starts usually above the age of 60, and it's thought that above the age of 80, 1 in 5 people have the disease. And unfortunately, there's no cure currently for this disease. And because of that, and also because these people are elderly, they need a lot of care and for the everyday activities. So the economic burden of this disease is huge on our society, to the point that just in US alone, we spend about $300 billion per year. That's the economic cost that the disease, for this disease in the US. So how does the disease actually start, or how does it happen in the back of the eye? So this slide shows you a schematic in the back of the eye. On the right side of the panel, you see kind of the healthy area of the eye, where you see mainly 3 cell types. On top, these are the photoreceptors, and right under the photoreceptors, this is a cell type that we're interested in called the retinal pigment epithelium, or the RPE, and underneath this is the choriocapillaries, or the blood supply. So this is the part of the eye that is towards the periphery, and that's at least healthy in those people with AMD. As you move towards the center, you see what is called the dead zone in the center of the eye, where you don't have any photoreceptor surviving, and of course no RPE is surviving either. This is an area where these people are completely blind, and that's why they can't see anything in the center of the eye. Now, in between the dead zone and the healthy zone, there's this area called the transition zone, in which, as you notice, that the retinal pigment epithelium, the RPE, is missing, but the photoreceptors are kind of still there, even though they don't look very healthy, and as you can tell, they're slowly probably dying, and likely not also functioning very properly, because RPE is required for many of the functions photoreceptors perform. So it is thought that because it is the death of RPE cells that leads to photoreceptor cell death in these people, if you could replace RPE in these people provide an RPE patch, like especially an autologous RPE patch, you could protect the overlying photoreceptors from dying. At least at that stage, stop the disease from progressing towards the periphery. And the idea is that if in the first few patients you can demonstrate proof of principle and safety of your trial or safety of your cell therapy product, then you could move with time towards more center of the eye and start and actually even stop the disease from happening. And that's kind of the idea that we are working on to kind of develop an RPE patch, autologous RPE patch from AMD patients to be able to transplant in their eyes. And this idea comes from previous work that has been done in terms of surgical interventions. So what surgeons have tried for a long time is that, like I said, the disease is a disease of the center of the eye. It's the macula that is affected. So what people do, or what surgeons have done, is to cut out a piece of retinal pigment epithelium or RPE from the periphery and translocate, so cut it out from this side and translocate it into the macula here, into the center of the eye, and leave it there. Now, as you can imagine, it's a complicated surgery. When they cut out the piece from the periphery, it leads to hemorrhage and other complications on that part, and often the graft does not integrate into the center. But in many cases, when the graft does integrate, surgeons notice that the survival was for several years to come, and it affected or it protected visual acuity in those patients for years to come. So for me, this is a very important proof of principle that an autologous RPE transplantation is feasible, that if we are able to manufacture an autologous RPE patch in vitro and don't have to do this complicated surgery of cutting patch from the periphery and moving it into the center, then we could clearly provide potential benefit to these patients. Hence, the idea that we're working on, which is that we start with the patient, their somatic tissue. We start particularly with blood cells, but you can start with other somatic tissues, reprogram those cells into iPS cells, and from those iPS cells differentiate them into RPE, develop a cell therapy, and treat those patients back with their own RPE patch. So we're working on a phase 1 IND application for this therapy to be filed with the with the FDA probably in a year or two from now. So when you work on a cell therapy product or any kind of therapy product, the idea is that you should start with the end product, which is going to be what is going to be your clinical product, and you work your way backwards. How are you going to actually manufacture that product in a GMP compatible fashion in a clinical suite, and how are you going to file for or perform all the preclinical work to file for your phase 1 IND application for such a cell therapy product. So this small video shows you our cell therapy product, our test article, so to speak, that we plan to transplant in patients. So this is a 4 by 2 millimeter patch of RPE cells on a biodegradable scaffold that a surgeon's assistant is manipulating to demonstrate pigmented cells on top and no cells or a scaffold on the back. So what I'll be doing is I'll walk you through the whole process, how we actually come about manufacturing this patch in an autologous fashion from AMD patients, and how have we optimized the whole process to work in a GMP compatible fashion. So as many of you may know, an IND, Investigational New Drug application, has 3 main elements. One is the manufacturing, called the CMC, Chemistry, Manufacturing, and Controls. This is the part where you actually describe how your manufacturing process works, what is going to be a source tissue, what are your control strategies, and what are your release specifications. And then the second main part of an IND application is the preclinical data, that is the animal toxicity and efficacy data. So basically you'll demonstrate that your cell therapy product, your test article, is not toxic in an animal species and demonstrate some efficacy in some kind of an animal model of for an eye indication. And then lastly, you need to have your clinical protocol to be able to transplant such a product into patients. So I'll start actually with the clinical protocol, just to briefly summarize this, because I won't go into much details of this today. So just again to reiterate the point that this is a phase 1 primarily safety and monitoring study. Basically we will transplant about a dozen patients, and we'll track events related to disease progression. Basically what we want to see is that the occurrence of disease, does it accelerate or does it slow down after transplantation of our patch? We'll monitor for events that are related to side effects in the eye that might be considered as complications of such a surgical procedure. Some of those are listed here, for instance, tear in the retina, hemorrhage, condition called proliferative vitreoretinopathy, cataract, endophthalmitis, and so on and so forth. So these are primary safety events that are very important to monitor in a phase 1 trial. And then, of course, we will have a data safety and monitoring committee as an oversight that will decide about events based on the event occurrence and at what speed we are going to be transplanting these into patients, how will we recruit a given patient at a given time, and then at what step do we move on to the next patient, and so forth for the whole trial. So before I start describing the manufacturing process, I want to kind of list out some of the manufacturing and regulatory challenges that are unique to an autologous induced pluripotent stem cell-derived RPE patch. As you saw, our patch is a combination product. So what that means is it has cells and a scaffold in it. And of course, for transplantation, we will need a transplantation tool. So it is by the criteria of a cell therapy, it is considered, or a therapy, it is considered a combination products of cells, scaffolds, and tool. And that means we will need to provide regulatory documents related to all three of these pieces of our product. Because it's an autologous product, we need to have, that means that we need to be manufacturing the cells from every patient every time for every patient. So that means we need a manufacturing process that is very efficient and very reproducible so that when we manufacture from one patient to the other, we do not have a process that fails. Of course, we want pure cells at the end. There shouldn't be any iPSC contamination because, as many of you may know, iPSC contamination in cells in your final product may lead to formation of tumors or teratomas or other cell type in the eye. And we need cells, patches that are functional so that they can go in, perform the functions RPE cells perform in the eye. Another important point to remember for such a process, for an autologous iPS process, is that the manufacturing process is extremely long. So we need to identify intermediate stages where we can actually stop the process, and if something fails, we can go back to those intermediate stages. And we need to identify go, no-go points so that we know whether the process is going in the right direction or is it failing, and if it's failing, what do we do, and do we go back to one of those intermediate stages. So with that brief introduction about our product, I want to start with our manufacturing process. So our manufacturing protocol to make mature RPE cells from iPS cells actually follows a very developmentally guided process. So what I mean by that is that the cells go from iPS cell to RPE to mature RPE cells following these various stages like neuroectodermal, RPE progenitors, committed RPE, mature RPE, all the way to mature cells in a very developmental fashion, as if the same stages, they follow the same stages as the cells do when they are inside an embryo, when they're developing inside an embryo. And we have been able to track that by looking at markers for every given stage at different time points. And the reason why this happens, that the cells follow all this developmental pattern for going from iPS cells to mature RPE, is that we have optimized the differentiation protocol such that this combination of different growth factors and the concentrations used at different time points allows them to really mimic kind of the developmental biology of in vivo developmental biology of human RPE cells in a dish to get to mature cells. And the process is highly reproducible. We get RPE differentiation from different iPS cell lines in a very reproducible fashion. For instance, I'm showing you an example of 4 different iPS cell lines. Is one with healthy fibroblast, iPS cells derived from healthy fibroblast, iPS cell derived from healthy blood cells, and iPS cells derived from fibroblasts of 2 different AMD patients. And in every single case, we're able to get, as you can see, RPE cells at a very, very high efficiency from iPS cells. And these black cells, these black things at the bottom of the dish, these are all RPE cells. We have a very simple enrichment step that purifies RPE cells to get to this pure RPE cultures, as you can tell here from this dish that is full of pure RPE cells. And by looking at different RPE markers by this flow assay, you can tell that more than 98% of the cells are pure RPE cells. So we can get pure cells, and we can get them at the protocol is efficient, and we can get them reproducibly from different patients and from different iPS cell lines. So now I'll just show you how we actually take these cells, and from these cells we make an RPE patch on a scaffold. The scaffold for us of choice is a degradable PLGA material, polylactic glycolic acid. These are electrospun nanofibers of PLGA. They've been optimized such that the cells would form a monolayer on top, and it would degrade in a desired time that we require for it to degrade. As you can see, the cells start to nicely proliferate on this PLGA patch. This is one RPE cell 3 days after seeding onto the scaffold, and it starts to interact with the scaffold fibers through these nice legs and arms that it extends out. And in about a week, the cells become nicely confluent. However, not all cells are polarized. As you can see, here's a patch of cells that is polarized, and we can tell that because RPE cells have this classical apical structures called the apical fibers, and they extend out on top of the cell. And you can see all of these cells in this group have those apical processes, whereas if you move further away, these cells do not have as many apical processes, or further away, these cells don't have any at all. So in 1 week, the monolayer of RPE cells is not fully polarized, but if you wait for 4 weeks, all the cells are nicely polarized on the monolayer, and they're ready as functional RPE cells. I'm going to show you some of the functional tests that we perform on these cells. So looking at the cells from a different angle, this is a side view using scanning EM. Again, you can tell these are the apical processes demonstrating that the cells are polarized. Apical melanosomes, tight junctions between neighboring cells, and here the scaffold fibers on which these cells are growing. And you can tell another interesting feature of these cells, which is the formation of basal infoldings on the bottom side. This is a very typical feature of native RPE cells that you usually do not see on cells growing in plastic dishes, but apparently the 3D architecture of scaffold provides this possibility for the cells to make basal infoldings on the basal side. So we've looked at the cells by multiple different ways, including several different gene expression for different markers. I'm showing you an example here only of 3 different markers, ZO1 in green, which marks the tight junctions. Phospho-ERM is a protein that is actually present in those apical processes and is responsible for functioning of those apical processes. RPE65 is an enzyme that is responsible for making visual pigment. So you can tell these 3 are expressed at a very high level, and we have looked at several hundred genes that are specific to RPE cells, and all of them are expressed at a very, very high level in these cells, so suggesting that these cells are very native-like RPE properties. We've looked at their function. One example of that is shown by this assay that we perform, which is called the fluid transport assay. Basically, RPE cells have the ability to transport the fluid that photoreceptors dump onto these cells. RPE cells transport that water from apical towards basal side into the blood supply. And the way that happens is that they have purinergic receptors on their apical surface. When ATP binds to those receptors, they're activated and they mobilize intracellular calcium stores, which activate the chloride channels on the basolateral sides. And as the chloride ions are moving from apical to basal sides, they drive isoosmotically, they drive water with them. And we're able to measure that water transport by taking that whole RPE patch. So now we're taking an intact RPE patch of about 10 millimeter diameter that we transplant in our, that we mount in our physiology rig, and with these 2 capacitance probes on each side, we're able to tell even the slightest water movements that happened from one side to the other. So in addition to measuring water, these capacitance probes can also measure the electrical properties of the monolayer, which you see in the bottom panel here. These cells, this patch has electrical resistance of about 200 ohms per square centimeter, and an electrical potential of about 2 millivolt. That is the difference in resting membrane potential of apical and basal sides, again suggesting that the cells are polarized. So on top you see that at steady state the cells are able to transport water at a very low level, around 1 0 to 1 microliters per square centimeter per hour. But when we stimulate them with ATP that drives this intracellular calcium stores, you can increase water transport, and then we can demonstrate the specificity of that by washing out ATP or by blocking these chloride channels to show that this water transport is specific. So now that I've shown you that we can robustly make RPE cells from iPS cells, from different iPS cell lines, and we can demonstrate that cells are functional, I want to show you that we have been able to optimize the manufacturing process of these cells that is GMP compatible. So this whole process we have mapped out from day 0 to day 157, so it takes about 5.5 months for the cells to go from patient all the way back into patient. So basically day 0 is when we draw blood from the patient, go through isolation of CD34 positive cells, making of reprogramming of CD34 positive cells into iPS cells, and mini banking the cells at passage 5, or making a working bank at passage 10, and then starting a differentiation at passage 11 of up to 3 clones, going through this intermediate stages of RPE differentiation to seeding on scaffolds, and finally getting a clinical product at day 157. So as I mentioned early on, that we need to have, for such a long process, you need to have intermediate stages where this process can be stopped, and you can go back to those stages if needed. So we have been able to identify 4 different stages, starting with the patient material, iPSC mini bank, working bank, and the RPE progenitor stage, where we can go back and we can cryopreserve cells at these different stages, and we can go back in case the process fails at any step, and we don't have to start from day 0 again. Also, to identify if the process is going correctly, we have in-process quality controls, including important ones as sterility at different time points. But at the iPSC bank stage, you have to kind of validate your bank in terms of the quality of iPS cells by loss of plasmid, karyotyping, that the iPS cells belong to the patient, so identity of cells, and perhaps sequencing for some of the selected genes to look for the quality of cells. And then the other assays include, in terms of quality check, is that the cells are differentiating correctly, looking for RPE markers at different time points. And then finally, identity and functional test and purity of cells in the clinical product. So I'll show you a couple of examples of some of these assays that we have tested on our GLP product during the manufacturing process. And those assays seems to work very well and does validate our manufacturing process well. So one of the main tests is the loss of plasmid in a working cell bank of iPS cells, and this is one example where we tested, as you can tell, almost 15 different clones from 3 different donors. At passage 10, in pretty much every case, we have lost plasmids from all of these cells at passage 10 of our working bank. So we know that our cells, when they go into differentiation, are free of any exogenous plasmids. Purity of cells. So this is a process, this is gene expression analysis of cells during the manufacturing process, starting from iPS cells through the intermediate RPE stage to mature RPE stage, and comparing them to primary RPE cells. And we're looking at expression of different iPSC gene or some other like fibroblast gene. And you can tell very quickly the cells start to drop the expression of iPSC markers, and in mature RPE cells we do not have any expression of any of this iPSC marker or of the fibroblast marker. Interestingly, the expression is actually even lower than the primary tissue. So the cells really do not express any of the iPSC genes at the mature RPE stage, and also perhaps do not have any contamination of iPS cells at that stage. Another gene expression analysis now specific to RPE markers doing at the same 3 stages of differentiation comparing the primary tissue. The expression of 2 of the progenitor markers like MITF and PAC6, they go up from iPS to RPE progenitor stage, shown here in yellow and black, and then as the cells mature they go down. Other genes like OCA2 pigmentation marker goes up as the cells start to become RPE and stays constant pretty much as the cells keep becoming mature, and then other mature markers like RPE65 or BEST1 slowly start to increase as the cells are maturing. And again, the expressions are very comparable to primary tissue. So again, suggesting that in this GLP manufacturing process, cells maintain the RPE phenotype and are very similar to native RPE cells in their qualities. Structurally, the cells look, again, nice polarized RPE cells. This is a pseudo-colored image of SEM image, scanning EM image of RPE cells on a scaffold. In blue, you see the scaffold fibers, and in brown are the cells and the nuclei that you see from the side. Because it's a side view, so you only see the cells and the boundary. And you can see 3 nuclei here, and then, but you see the apical process pretty much of the whole monolayer, and you can see all the cells in the monolayer have very extensive and very homogeneous apical processes, suggesting that all the cells are similarly polarized in the whole monolayer. The cells behave very similar, as I said, again, structurally and electrical properties to primary tissue. Is an example of comparing iPSC-derived RPE on a scaffold to primary human RPE on a scaffold. And you can see by scanning EM, again, they look similar. By electrical resistance, the cells look very similar. They have very similar electrical properties of the monolayer. One more example, looking at electrical properties of cells on a scaffold. Now we're comparing here iPSC-derived RPE on a transwell, which is a plastic membrane, versus the PLGA scaffold. And as you can tell from the 2 graphs that they're very similar to each other. The assay is basically looking at the ability of cells to hyperpolarize in response to reduced potassium concentration in the apical band. And it's a physiologically relevant assay because when the light shines on photoreceptors, they shut down the potassium channels. That reduces the potassium concentration in the apical band. RPE cells respond to that, they hyperpolarize and they correct the potassium concentration. And the opposite response is measured when ATP is added and the cells depolarize because of the change in chloride concentration on the basal side. So we can measure all those properties, and as you can see, the cells really respond similar to native RPE cells and similar between transwells and the scaffolds. So that's more about function, gene expression and function of these cells, but then what about the purity of cells? So what we did is we did an assay to demonstrate whether there are any iPS cells in a final product or not. So what we did is we actually spiked purified iPS cells into our final product, into our RPE cells actually, and seeded them all onto scaffolds. So the assay here is that we have either 100% RPE cells or 100% iPS cells, or in between where we have 99% RPE plus 1% iPS and 90% RPE plus 10% iPS. And then we seed everything onto scaffolds. So this is the last stage of our manufacturing process. And we look for OCT4 and TRA181, 2 of the iPSC markers, after the cells are seeded onto scaffolds. As you can tell on day 0, of course, we can detect in 100% or in 10% some of the iPSC cells, even in 1% we can detect iPS cells. But very quickly from day 0 to day 2, almost by day 7, and really by day 14, we do not detect any iPS cells in any of the mixtures, spiked cells, or pure iPSC population, suggesting that iPS cells, even if they are forced to contaminate the RPE cells, they do not survive in our culture conditions on our scaffolds. So this is very encouraging because, you know, I showed you data before that iPS cells are not present in our differentiation process, and second, I'm showing you now data that even if they were, even if you force them to be there, they do not survive in our culture conditions. So that makes us very confident that there is going to be no iPSC contamination in our final product. So we also have spent quite some time characterizing our scaffold, how it degrades, and we have followed its degradation in vitro and in vivo. I'm going to showing you one example here of in vitro degradation. So this is scanning EM of scaffold fibers from day 0 to day 49, and as you can tell how the scaffold fibers are slowly kind of melting together and degrading away. This is a stage that when we transplant them in animals, so at this stage they kind of pretty much like a little sheet or a mat of fibers. If we look at them from a side view, again by scanning EM, that tells you that the scaffold is getting thinner over time. So here, if you pay attention to the scale bar, the scaffold is about 10 micron thick on day 0, and at day 63 there's about only 2.5 micron thickness of scaffold is left. It's still quite porous, it's still hydraulically nicely conductive, so we don't think it impedes in the nutrient flow, but it's a very thin piece left, and within another week or two most of it is going to degrade in the eye. So now I'm going to switch to, for the last few slides, switch to preclinical studies and share you some of the preclinical data that we have developed as part of the feasibility studies, which is guiding some of the GLP work that we're just starting to plan out for the next few months. So the first experiment is done in immunocompromised rats, because to be able to monitor long-term toxicity and tumorigenicity of these cells, you have to transplant them in a species where they can actually survive long-term without immunosuppression. And that's why we chose to use immunocompromised rats. And as part of this feasibility study, we're looking at survival of RPE cells, iPSC-derived RPE cells, either day 1 after post-injection or a month after injection. And you can see in both cases, the cells just sit in the subretinal space. They kind of clump together, but they're nicely surviving in the subretinal space for a month here, and not leading to any overt looking structures. And this forms the basis of the study that we were planning to do in terms of looking at the long-term tumorigenicity and toxicity. As a positive control, if you inject iPS cells in the subretinal space, these cells lead, as expected, to this massive tumors, or actually teratomas, in which you can actually identify different lineages from alveoli to tubules to ovarian follicles, glands, and cartilage. So we know that at least our cells, our RPE cells, are not forming any of these tumors. Only pure iPS cells are forming this kind of tumors in the eye. The second model that I'm going to switch to is also a rat model, but it's called the Royal College of Surgeon rats. This is a genetic model in which there's a point mutation in a receptor called MERTK that is responsible for phagocytosis of outer segments. And because of these, RPE cells in these rats are not able to phagocytose photoreceptor outer segments, and hence the retina starts to degenerate and these mice go blind. So what we did is we transplanted iPS-derived RPE cells in these rats, and at day 28, and looked at them at day 100. You can see at the site of injection you have two nuclear layer that are intact. So these are the two types of nucleus that are present in the retina. These are the outer layer is the nucleus of the photoreceptors, and these are the nuclei of the retinal neurons. But if you look at the site away from the site of injections, you can see further away as you move away from there, the nuclei of photoreceptors are already all dead, and in this side there's no photoreceptors left. So our cells are able to protect overlying photoreceptors from dying at the site of injection. If you look at the close-up of this, you can see nicely here human cells in green and RPE cells co-labeled in red, and they're able to protect photoreceptors right on top of them. And as you move further away from the site of injection, the protection starts to diminish, as you can see by the thickness of the nuclear layer of photoreceptor. We perform functional tests in these rats, and the functional test is called optokinetic tracking. Basically, the rats are put on a platform, and there's a cylinder moving around them which has black and white stripes, and because of these the stripes, the rats start to track the stripes if it can see. But if the rat can't see, it won't track the stripes. It will just randomly look on both sides. So, and you can kind of quantify how much time the rat spent tracking the stripes or not, and that way you can tell if the rat could see or not. So, of course, here you can tell in this graph, the wild-type rat can pretty much see all the time. The dystrophic rat, which did not get the cells, it slowly starts to, the retina starts to degenerate, and over time you can see it doesn't see anymore for up to 300 days till the measurement was done. iPS cells were injected in this dystrophic rat at day 25, and then we did two measurements at day 60 and day 100, and you can see by day 100 the rats that got our cells could pretty much see as good as the wild-type rats, at least on this test, suggesting that these cells have ability to protect the overlying photoreceptors from dying in this setting. However, I want to go back to the model where I started, in which I suggested that we want to transplant an RPE patch. So what we want to have a model where we can actually transplant an RPE patch and see how the patch integrates in the back of the eye and how it protects the overlying photoreceptors. Now, unfortunately, to do that, you cannot do this kind of studies in rats easily, because to transplant a 4 by 2 millimeter human-sized patch, it won't be feasible in rats. So we needed a large animal model. So we decided to use pigs for this reason, because pig eye size and eye anatomy is very similar to human eye size and eye anatomy. So we could test the same size patch that was going to be tested in that is going to be used in human could be tested in pigs, and we could test the transplantation tool in this and the surgical procedure that we want to use for human and these pigs. So first of all, to be able to transplant such a patch, we had to develop a transplantation tool. And this slide shows you the tool that we have come up with. It's a very similar cannula, very simple cannula, where the transplant is actually inserted in the tip here that you see in the high magnification front top view and the side view of the cannula, and the scaffold is moved around just by using hydraulic, and by a foot paddle the surgeon is able to control how the transplant moves in and out of this cannula. So back to the pig model, the model has, because in pigs there's no genetic model of RPE cell death, we came up with an injury-induced RPE ablation in this model. So basically we use micropulse laser and ablate RPE cells in the back of pig eye. On the left you see infrared imaging showing laser damage of the RPE in the back of the eye. You see this kind of a window defect. It's about 8 by 4 millimeter window defect in the back of the eye. If you look at that by histology, you can see RPE cells are clumping because they absorb the laser heat and energy and kind of basically burst out and then clump. And as a result, photoreceptors, these are the overlying cells, they started to die. So the idea here is that now we come up with our patch and put it right here and protect those overlying photoreceptors from dying. So I'm going to show you a movie how we transplant our RPE patch in these pig models. So the surgeon has already prepped the eye here. You can see now he's making the incision bigger, and in the next frame he's going to show you that the cannula is going to bring the cannula inside. Here's the RPE patch. Here's a part of the retina that has been lifted called by subretinal detachment. Here's a cut in the retina that has been done. He's aligning the cannula, and it's going to insert the patch right under the retina right there. And he's going to kind of adjust so that the patch stays under the retina, so that, and then the retina is flattened so the patch would eventually stay there forever. So you can then monitor how the patch survives in the back of the eye. This is one example looking in those. This is actually an empty PLGA scaffold. We're just looking at how the scaffold degrades in the back of the eye, and does it cause any damage to the eye. Here we are comparing 1-week-old scaffold versus 6-week-old scaffold, and you can tell by the thickness of the patch. And by the way, this is images taken by a technique called OCT, optical coherence tomography, which looks at reflectance from the back of the eye. This is a whole retina. You can see by this technique, and then underneath the retina you see this patch here. It's quite thick at 1 week, about 10-20 micron. Whereas in about 6 weeks, the thickness has dramatically decreased because the scaffold the PLGA is degrading slowly, and the cells would eventually integrate into the Brooks membrane. So if you do that with the scaffold with cells, again same view, you can see the scaffold underneath the retina. This is a cut view, this is a top view, this is a scaffold, this is a site of surgery. Now, to be able to do functional measurements of the scaffold, what we do is we use a technique called multifocal ERG. Basically, this technique measures electrical responses from the back of the eye, but depending on light pulses that are stimulating different areas of the eye. So we're shining multiple pulses, and depending on where the light shines, you get an electrical response from that part of the eye. So for instance here, if the light shines on a healthy part, you get this nice, robust electrical response. If the light shines on the lasered part, you do not get any response. And if the light shines on the transplanted part, you get a response that is in between the injured and the healthy area, suggesting that at least in this part that you see here, the RPE cells are able to protect overlying photoreceptors from dying. We have confirmed these findings by histological analysis of the cells. Here in the top panel you see green is the human RPE cell patch, and in magenta you see the photoreceptor outer segments. So it's showing you that they're nicely protected, and the RPE patch was able to slow down the death of these photoreceptors. You see this nicer also on the lower panel, where we are comparing in the same eye laser area that did not get any transplant to laser area that got the transplant. And here's a transplant. You can see in the laser area there's no transplant here. But if you now compare the thickness of this nucleus layer of photoreceptors, you see it's much thicker, almost double the size of this area which did not get the transplant, suggesting the protection of photoreceptors caused by RPE patch, specifically in this area. So that brings me to the end of my presentation. This work that is being done in pigs is now has formed the basis for the pig efficacy study that we're doing in-house. And with that, I'd like to thank the whole team that has contributed to this work. I should say that there's a very large team that is involved in doing a lot of this work, starting from, as you saw, from the lab work to GMP grade manufacturing of cells, to functional testing of those cells, and then different animal models. So this is an effort that, as I say, it takes a whole village to do it. So I want to thank the whole team, every single one of them, and I'll be happy to take any questions.
Thank you very much. That was an incredibly interesting presentation. If you do have a question for Kapil, please type your question into the question pane of your GoToWebinar window. We do have a couple of questions lined up for you. So we have a question from Abbas Raza, who I'm just about to unmute him. Hello, Abbas. Can you hear me?
Yeah, hi. I can hear you. Hi. Can I ask the question number?
Yes, please go ahead.
Yeah, hi. Hi, Kamal. That was an excellent talk, and I'm from University of Minnesota School for Drug Design, and I'm wondering if this therapy can have implications in patients with livers. Yeah.
Thank you. Thanks for your comment and your question. Yes, and I don't want to say no. The answer is yes, but I was hesitating because for disease like livers, especially the kind that affects the RPE, most of those diseases are monogenic diseases. So you would still have the genetic defect in the cells because you made them from the same patient. Unlike in AMD, where it's a polygenic disease and there's no mutation that directly affects the disease in Leber's and other forms of retinitis pigmentosa, the cells would be still affected. So one would have to actually do gene therapy on iPS cells, and it's called ex vivo gene therapy. So technically it's possible to do, but I think the field is not quite there yet. The gene therapy field is kind of independently going on and in parallel trying to create gene therapy for different diseases. And I think hope is that over the next few years the two fields will merge and we will be able to do gene therapy, ex vivo gene therapy on patient-specific cells, and then differentiate them into any cell type and be able to do transplantation.
Excellent. Thank you very much.
Thank you very much, Abbas. So we have another question from Mehdi Safa. So I'll just unmute you now. Mehdi, are you there? If you unmute yourself, Mehdi, then nope, doesn't look like. What I will do is ask a question for you. She says, are you intending to do a whole genome analysis to rule out any genomic instability, mutations, etc., to evaluate the safety of the therapy in the long term?
Yes, so what we are doing currently is we're doing two things. Of course, we're doing karyotypic analysis at the working bank stage of iPS cells to look for like large-scale genomic instability. And what we do is, and what we have already done with our GLP bank is a very thorough, very deep sequencing, exome sequencing of the oncogene panel, which is of bigger concern, and looking at if there's any mutation, comparing that to the primary tissue of the patient itself. And that will lead to hopefully to a more of a tighter quality control of the cells that we're making, so that we don't end up transplanting cells that have either large deviations or large changes in the genome, or any mutations that could potentially be tumorigenic in those cells.
Thank you very much. So we have another question from Fernando Lujudis. I apologize if I've mispronounced your name, so I'll just unmute you now. Fernando, are you there?
Oh, yeah.
Hi, would you
Can
like
you hear
to?
me?
Yes, I can. Would you like to put your question to Kapil?
Okay. Thank you, Dr. Braff. It's incredible talking. I would like to know what kind of tests do you make on the scaffold before seeding? I mean, how to evaluate the reproducibility of the scaffolds or even the clinical products?
So the scaffold is just the scaffold material itself. So we test the reproducibility of the electrospinning process by imaging. We have performed extensive scanning EM of the scaffold, and then we have developed a software that kind of analyzes those pictures and looks at the thickness of the fibers, the pore size, the pore diameter, and everything, and provides a matrix of what is the quality of all of those parameters on our scaffold. And using that, those two, SEM and image analysis, we're able to tell how reproducible is our scaffold. And fortunately, we have a process of manufacturing of scaffold that is highly reproducible.
Okay, thank you.
Thank you very much, Fernando. We also have actually a question from Scott Schachtel. Scott, are you there?
Yes, can you hear me?
I can. Go ahead.
Okay, very good talk, Kapil, thank you. I was just wondering, how closely did you work with the FDA in defining the qualifying criteria for your preclinical and clinical data, and how was that process?
So, for biologics, you have the possibility of interacting with the FDA at least on two different occasions. One is called the pre-IND and one is called the pre-IND. So basically, you have to summarize your process and send it as an application with a meeting request, and then you get your responses back to some of the questions that you've raised in that application, and then you get to have either face-to-face or telephone conversation with them for an hour and go over some of those questions. So we got a chance to talk to them, and we also had a follow-up conversation. So their input was very critical, and is now being very helpful in kind of designing and finalizing our preclinical studies that we are currently doing.
Great, thank you.
Thank you, Scott. Actually, I also have a question. So obviously the productions of the cell and the scaffold is a very long and technical process. What would you say the biggest challenges you had in converting that process from a purely research one to one that could withstand the requirements of a clinical trial? Obviously there were many. I just wondered if there was like a theme or one in particular.
I think, yeah, no, there were many. I think one of my favorite one that, actually two, but I'll pick one for now, is when we culture these cells in the lab, everyone uses trypsin, standard trypsin EDTA, to pick the cells and further seed them. When we went into a protocol that was going to be GMP compatible, we realized that trypsin could not be used anymore, and we had to use a substitute that was compatible with GMP. And just because of that step, that we had to change significantly when to pick the cells from dishes and what stage to reseed them and all. So it actually had a major impact on our manufacturing process, just the choice of trypsin, as trivial as a choice of trypsin.
Did you ever have any problems about sourcing the right reagents that could be used in that you'd been used to using in research use and then moving into clinical or preclinical use? Did you have any problems sourcing any of the GMP reagents, for example?
Not really. I mean, there are a couple of reagents that were not available as animal-free, but we are working with those companies to have animal-free reagents available. Especially it's for biologics. Wherever these are biologics, it's always an issue. So, including working with R&D and with another company to create some reagents that are more animal-free.
Okay, thank you. We have one more question from Mandeep Singh. So I'll just unmute you, Mandeep. Oh.
Yeah, hi. Can you hear me?
Yes, we can hear you. Please go ahead.
Hi, Dr. Bharti. It was very wonderful, this interactive session. And I just have I'm just curious about one thing. Listening to what you've covered with the iPSC, you've taken care of that part largely. So were you able to look at the perspective from a development of a cancer stem cell, or use a marker, such as CD133 to look into some kind of development on that front? Just out of curiosity.
Not sure what you mean. So you mean in our cells look for CD132?
133, yeah, something like that. Any of the cancer stem cell markers, if you were able to look at that, I can see that you've largely taken care of the iPSC markers, and to find out.
Right. No, yeah, but so, is there any particular reason why this marker is interesting? Is it known for ocular tumors, or I'm not sure. I'm not aware of this marker.
Not in particular about ocular tumors, but in general, this is a kind of a marker for cancer stem cells. So we do know that there can be a possibility for development of a cancer stem cell if there is a genetic mutation during the process.
Mm-hmm.
I'm not sure if that is possible, because since I don't know the culture techniques that you've been using and all the experimentation has been so far.
Right. So if you take cells that are positive for CD123 and inject them in rats, would they lead to tumor formation?
Absolutely. Yeah. Okay, I mean, this is what we know about CD133 so far.
Right. Okay. So I would say we've done it indirectly. We haven't looked for CD123, but we have, as I showed, data that we have injected our cells into immunocompromised rats, and we do not see any tumor formation in those rat eyes. So I'm fairly confident that we won't have any expression of CD123, but thanks for the suggestion. We'll look into it.
It's actually CD133, 133.
Okay, sorry, couldn't hear it. 133, all right.
Anyway, thank you. This was wonderful.
Thank you very much, Mandeep, and thank you for your question. We're nearly up on our time right now, so if you do have any other questions, please feel free to forward them to the contact address for this webinar, and we will forward them on to Kapil. The webinar is being recorded, and we will be sending you a link very shortly to where you can watch the webinar again if you should so wish. So all that leaves me to do is to thank Kapil very much for a great presentation, and thank everybody for joining us today. So have a great day wherever you are in the world. Thanks very much. Bye-bye.